All right, so I had talked about the different types of polyunsaturated fats last time, and we had talked about that omega-6 to omega-3 balance that you want to have in your diet, and I touched on the weakness of nutritional labeling a little bit, and how the way we describe things in nutritional label isn't really telling you the information that you ideally want to know about the fats that you're consuming. So what's the big deal here? I talked about this slide a bit and how the kinks in the double bonds, the kinks in the fats due to the double bonds prevent ideal packing. All right, so then I want to make this point. I didn't make it very well last time. The reason the melting temperature is higher in fats is because if you look, as opposed to oils, if you look on the left, in these saturated fats, there's a lot more overlap between these chains, right? So I kind of, if you go back and forth between me highlighting and unhighlighting the interactions, wherever there's like a yellow highlight between the fat chains, that's hydrophobic interaction. That kind of hydrophobic interaction is going to have to be what gets overcome to liquefy a lipid that has congealed. Whereas on the right hand side, where these kinks, there's significantly less hydrophobic interaction per square area, surface area of a surface of lipid. So that lipid that has congealed there is going to have a much lower melting temp, and so that would be the solid form of it there. Going from that to a liquid where the fats are just like a bowl of spaghetti is going to be at a much lower temperature, all right, because there's fewer hydrophobic interactions to overcome. It became clear that some of you may not have understood that at the end of last class period. So then I guess I just wanted to go back and reiterate that point. And I had shown this, showing that correlation between saturated fats and versus unsaturated fats and the relative melting point temperature. All right, so I'm not gonna reiterate this too much. I guess that you guys have to tell me where I stopped. I forgot. I talked about this too. So I talked about hydrogenating fats and how we can take double bonds and catalytically break them and add hydrogen gas across them using these nickel, palladium, or platinum catalysts. One of the downsides of this process is sometimes we can take cis fats and isomerize them to trans fats, and that's a problem because trans fat is going to look, trans fats are going to have the same conformation as these saturated fats on the left, but it's going to be even worse because they'll be like locked into that position. Saturated fats still are free to move if they want, and so those fluids are a little bit more, those fats are a little bit more fluid. A trans fat is obligated into that extended close pack form, right? And there's other sort of more complicated reasons why trans fats are bad in terms of biosynthetic catabolic pathways or anabolic pathways that we're not going to go into. But oh yeah, this is where I left off. Heart disease. Now I remember. Yeah. And you had been asking me about Arizona. Was that you? Yeah. I don't know. I don't have an answer for that one. I wish I did. Maybe it's a cultural thing. Maybe it's the kinds of foods they eat down there. I don't know. Yeah. Well, it's roughly it's roughly the Rocky Mountains. And then a little patch through Minnesota as well. But the Deep South, the Midwest, and large portions of the East Coast are all pretty blocked up right now. The American diet. All right, so this is where I want to start. Triglycerides. Up until this point, we've been talking about fatty acids, the most fundamental, the simplest way that we package fat. We're going to talk about a few others. I'm going to talk about triglycerides today. We're going to talk about glycerophospholipids, and we're going to talk about lipoproteins. These are three other categories of fats that's important to understand in your nutrition and food intake, your utilization of fats. What is a triglyceride? Well, it's really easy. We just take glycerol, also known as glycerin. Has anybody ever used glycerin at all by itself? You've certainly all used it because it's a component of like lip balm and all kinds of cosmetics. It's in a lot of stuff. But sometimes you can just go to the store and buy pure glycerin or glycerol and use it. Some people use it for really dry skin in places. You can stick a little bit of it there. It's a common component of hand creams. Well, anyways, you take glycerol, which is this 3-carbon chain. It says 3-carbon alcohol. Each of the carbons in the 3-carbon chain has one hydroxyl group on it. And the hydroxyl group on the glycerol, that alcohol group, reacts with the carboxylic acid. And I don't know, maybe last week, I don't remember exactly when, but I did go to the board over here and talk about the formation of esters. I think maybe Jake asked me what an ester was and how that formed. Ester is a type of a chemical functionality that is formed by the reaction of a carboxylic acid and an alcohol. And that's what we have going on here. So we have the acid on the end of the fatty acid, the carboxylic acid, reacts with the alcohol on the glycerol, and it's a condensation reaction. Condensation meaning water condenses out of the molecule. So I've highlighted in yellow the components of the water that are going to come out, and the remaining oxygen in that alcohol of glycerol acts as a bridge between the fatty acid and the glycerol. And so then that structure that you see on the right-hand side where there's C is double bonded to the O and then to an oxygen that is a bridge to the carbon backbone of the glycerol, that's called an ester. That's an ester linkage. And I think I had indicated that esters are often compounds that are odorants. They have a wide variety of smells to them. There are all kinds of different odor-containing compounds, odor-eliciting compounds have ester groups in them. Well, there is a triglyceride. And that particular triglyceride has the 3 carbons along, the 3 chains along the backbone, the 3 fatty acids, pardon me, are each 12 carbons long. It doesn't have to be that. It doesn't have to be that. Those chains can be of different lengths. Each of them can be different. Some can be saturated, some can be unsaturated. We'll see an example of that in a moment here. Right now, in fact. There we go. So they can have, here's a triglyceride where there's 3 totally different chains on there. The first one, fatty acid 1, is a polyunsaturated fat. There's two double bonds in that chain. That's a 1 plus 4, 5, 6, 7, 8, 9, 10, 11, plus 7, 18 carbon chain right there. The one below it is only a 16 carbon chain, fully saturated. And the one on the very bottom is a mono unsaturated, single double bond, single alkene group. And there's what, 8, 9, 10, 11, 18 on that chain as well. Okay. The one on the bottom would be an omega-9 fatty acid. Fatty acid 3 would be an omega-9. The fatty acid 1 would be an omega-6 fatty acid. And then the fatty acid 2 is saturated, so there's no omega rating for that. You guys follow that? Okay, learn a lot about fats here. So we can break them up too. So glycerides are really convenient little packages. It's like it's like a mini six-pack, a three-pack of fats. It's just a convenient way to store them all together. And the specific properties that a triglyceride has, and when I say property, I'm talking about biophysical property, like how they form fat globules, how they form micelles, how they go into a membrane, the effects they have on a membrane, like the fluidity of the membrane, the permeability of the membrane. All of those biophysical properties of the triglyceride is dependent upon the fatty acid, the specific fatty acid components that you're going to find in that triglyceride. So not all triglycerides are made the same. There are literally thousands of different triglycerides, although the actual number that occur is rather small. Okay, there's like it's like a Bell distribution. You know, there's a few that are highly represented and many other lesser types of triglycerides that you'll find. So you can break them down in a number of different ways. The first way is just chemically, and you can do that, the chemical breakdown, in one of two ways. The first is acid hydrolysis. So I have this, I have it going with H2SO4, which is sulfuric acid, but I could just as easily have put hydrochloric acid, which is stomach acid, up there. So when you eat a triglyceride, hydrochloric acid is happily able to cleave that ester bond and reprotonate the oxygen in the glycerol, and then to add an OH group onto the carboxylic acid and reconstitute the glycerol and the three fatty acids. Just going backwards. Acids can make it happen, and you can go backwards from a triglyceride to three fatty acids and glycerol. Another way of doing that, does anyone want to guess another way to do it? Get it right, you'll be squeaky clean in my book. Oh man, I'm giving you clues. Yeah, soap. Okay, good. Exactly, soap. What about it? Does anybody see the movie? I'm not necessarily advocating it, but if you've seen the movie Fight Club, you may have a better ability to answer this question. What happened in that movie, Damon? It seemed like you saw it. Yeah, mad fighting, right. But think about the non-mad fighting part of the movie. What else happened in that movie? He did make nitroglycerin. He did make nitroglycerin. This is true. And that was with the glycerol portion, the nitrated glycerol portion. That's nitroglycerin. And they did make bombs with that. But he also made, the other half of it was the soap. He didn't actually make the bombs out of the soap. He made the bombs out of triglycerides that he got from a liposuction clinic. Right? You remember that? Throw your mind back. He jumped in a dumpster, hauled a bunch of bags of triglycerides from a liposuction clinic out of the dumpster, poured a bunch of pool cleaning acid, which is nitric acid, all over it, and that's how you do it, and did what's called saponification. Saponification. Very old chemistry here. Very old chemistry. Yeah.
So is this kind of like, I've heard that to make like bars, is that sort
of... Exactly. Lights go on. Yeah, this is how you make soap, and this is why you use animal fat to make soap. This is why we were able to use egg yolk as an emulsifier, as a detergent, as a surfactant, right? Because they are amphipathic like this. So yeah, in the process, you take the triglyceride and you expose it to a strong base. The base I'm talking about here, if you want to make soap, you can use sodium hydroxide. Sodium hydroxide will give you the sodium salt of the fatty acid. It's now no longer an acid, right? It's the conjugate base. It's a carboxylate group. It's not an acid, carboxylic acid. It's the carboxylate group. It's been ionized, right? The proton, so an acid is a proton donor. The proton has been replaced, it's been ripped off, and it's been replaced with a sodium ion. So these carboxylate groups are really excellent soaps because now that it's charged on the end, now that the OH has been ionized and the hydrogen's off of that oxygen is negatively charged, and it's made that end even more polar, really, really polar. And so it's more amphipathic than it would have been as a fatty acid, and it's very good as a detergent, all right? Very good at emulsifying a greasy, fatty dirt or what have you. So this is done with sodium hydroxide, and you can reconstitute the glycerol on the other side. So in Fight Club, they used nitric. What they weren't using? They were using lye. That's what it was, not pool cleaner, lye, to do this same thing. And that's how soap was traditionally made. Lye is an extremely caustic, alkaline, highly basic compound that you can do the same thing with right here. So sodium hydroxide, that's Drano. You could take Drano and a bunch of animal fat and make your own soap. Or it doesn't have to be animal fat. You can use olive oil is a very common one. People have olive oil soap, all kinds of soap. You have like milk soaps, right? Goat's milk soap or something like that. Like you take the milk fat from goat's milk or something, and you can make soap out of that as well. This is how it happens. The process is called saponification. All right. So these are triglycerides. Really convenient way to package fats. We're not going to go into like the biology of them too deeply, although the deep biochemistry of them, although that's pretty interesting. I'm just going to talk about how your body absorbs them. All right, and I'm going to give you an overview, and you're going to have a fair bit of chemistry to be able to understand how the body absorbs this. So the basic flow here is that you're going to eat some fat, or you eat some food that has fat in it, and the fat is going to find each other in the crowd of your gut and form a fat droplet, all right? So you've eaten some food, and fat is mixed in, and it gets churned up in the, you masticate the food, the acids work on the food, the enzymes, and the fat has been liberated in this aqueous solution of the chyme. It's called chyme, sloshing around in your stomach and intestines, and we form fat droplets. These big fat droplets, kind of like if you had salad dressing, oil and vinegar, and you shook that up, and you saw the little bubbles of oil there. The next thing that happens is your body releases bile salts, and we're going to find out what that even means, what bile is in a bit. But the bile salts are going to break up those clusters of fat and essentially emulsify it. Bile is an emulsifier. It's also an emulsifier, amphipathic molecules. Then there are going to be some enzymes called lipases and lip, referring to the lipid, and ACE, meaning something that's going to cut, or an enzyme that's going to work on cutting them. So the lipases are going to hydrolyze the triglyceride. That word hydrolyze is the opposite of a condensation reaction. It's using water. Hydrolyze means to cut. So it's using water. These enzymes use water, and they use the water to cut the fatty acids off the glycerol in the very same way that I showed you that acids or bases do it in the previous slide. So the lipases will hydrolyze the triglycerides to fatty acids and monoglycerides, and then your cells, which are called the enterocytes, we'll see that in a bit, the enterocytes of the gut absorb these free fatty acids. All right? All right, so we got to get a couple foundational concepts in before I get into depth on how we digest triglycerides. The first is we have to figure out how to emulsify the triglycerides. That is, how do we take them from that big fat droplet, that enormous macroscopic bubble of fat that's in your gut? Actually, they're small, but you can see them. How do we take that big bubble of fat that enzymes cannot penetrate, they can't work on? How do we get them into an emulsion where they're teeny tiny microscopic droplets of fat? And then when that happens, we're going to have to get the enzyme to hydrolyze the triglyceride into the monoglycerides and the free fatty acids. So the emulsification process, there's two ideas that we're going to have to introduce. We're going to have to introduce the concept of cholesterol, what cholesterol actually is, and related to that is what bile salts are, because bile salts are made from cholesterol. This thing sitting over here on the table in front of us is cholesterol. Any of you are free to pick it up and stare at the wonder of the cholesterol molecule. It's a pretty incredible structure to have developed. And then the other concept we'll have to visit in talking about emulsification is what are called glycerophospholipids. An example of which is lecithin. We used lecithin in lab, didn't we, as an emulsifier? Okay. So first, cholesterol. It's sort of the mother molecule of a category. It's a mother molecule of a category of compounds called sterols. And sterols are themselves a type of lipid. They are made out of fatty acids. Well, the carbon comes from fatty acids originally, but in truth, the carbons that make up cholesterol are made by slowly adding two carbons from this molecule called acetyl-CoA. And we aren't going to get into acetyl-CoA too deeply here. It is a coenzyme. But we will look at acetyl-CoA more when we discover how our cells make energy out of food. So that's coming up in another week or so. We'll come back to acetyl-CoA. But acetyl-CoA is where the carbons come from. The last two carbons in acetyl-CoA, add those carbons on two at a time to build up this really crazy complex structure. Take a look at that, please, somebody. It took me and my daughter so long to make it. It's an interesting structure. And it'll become, the actual structure of it is going to be interesting to you when we begin to talk about bile salts. All the cells in your body make this. In fact, all animal cells make cholesterol. So the genes for making cholesterol are fairly highly conserved throughout the animal kingdom. All right. Cholesterol is the mother molecule for making all kinds of steroid hormones. So all steroids have this structural motif that you see on the board there, up in the upper left-hand corner. There's this four-ring structure, and you can see it in the body of the molecule there that you're holding. These four rings, 3 6-membered rings, 1 5-membered ring, and then this long isoprenoid tail, which is cholesterol. The different steroids that can be made from it include all the androgens like testosterone, progesterone, estradiol. Those are all the different molecules that are related to gender, the androgens. Then there's aldosterone and cortisol. Cortisol is an anti-inflammatory hormone. Aldosterone is related to the kidney function and water balance. But there's all these different steroid hormones that are made from cholesterol. So cholesterol itself is extremely important. This is why all the cells in your body are able to make it. One of the reasons. Another reason is that cholesterol is an essential structural component of all animal cell membranes. So it helps the membrane to maintain its structural integrity. Oftentimes, when you have unsaturated or polyunsaturated fats embedded in your membrane, you saw that there were these gaps, right? Well, you got to plug those gaps somehow, and cholesterol is a great way to do it. It also helps maintain the fluidity of your cell membrane. So your membrane, don't imagine it's like packing, like if you were to just bring as many people into this room as you possibly could, and we'd all be like sardines in a can. It's not like that. It's like sardines packed in a can that can move about a little bit. All right, so the lipids are free to sort of switch and move in a fluid manner about the surface of a membrane. Membrane dynamics is a really fascinating and complicated field that we're just going to leave right there. But cholesterol is important for maintaining fluid dynamics. And when you see this molecule here, you see there's a little circle sticking up. What part of the molecule would you guess that is there, Jake? You've got it in your hands. So map that mess that's in your hand right there onto the little yellow cartoon model that I have. I have this lipid bilayer. The pink balls are the polar ends of some lipids, and the green tails are some fatty acid chains, and then we have cholesterol embedded in the molecule. What part? And so in that you see the four rings. And there's this little circle. It's not a polygon. It's a circle sticking up towards the top. Why is the circle sticking up to the top? And what do you think on that molecule that's meant to represent? You can do it. Help him out, someone. Think deeply. Look at that molecule in his hands. Turn it around so they can see it all, model it. What do you guys think?
What part of it?
So,
Yellow.
yeah, this is cholesterol. And I'm asking specifically, what is this little round circle? Because they have this round circle always sticking towards the outside of the membrane. What do you think that is on that molecule you're holding? Yeah, it's the red one. What is that red thing?
It's not.
It's an oxygen and a hydrogen. It's an alcohol group. That's an alcohol group. So that's polar. That's the only polar bit on cholesterol. Just a tiny little lone hydroxyl group sticking up at the end. Yep. Yeah, exactly. And so you can see that there's this top rigid body, right? This framework, this rigid framework, and then there's this isoprenoid tail down there that's sort of wagging. So as you're holding that, the part that you're holding is not really moving, but the tail is wagging down there, just like a typical lipid tail. Yep, your cholesterol's happy. The tail's wagging. All right. So because all animal tissue can make cholesterol, all animal products have cholesterol. All right. So here are just some examples. You've got eggs. Eggs obviously have some cholesterol. The yolk is pretty much nothing but fatty acids and cholesterol. Beef, I got some liver over there. I don't know if any of you eat liver. It's good for iron. Cheese, muffins. Gosh, look at all that food. Yeah, ice cream, lots of cholesterol. Cholesterol abounds. Anything that has animal fat of any sort in it is going to have some cholesterol. All right. So, thank you. Good. I should have made. I didn't have enough molecules actually to make a bile salt and a cholesterol, and I thought it was better to have cholesterol. But another thing that cholesterol is able to make is a bile salt, and bile salts are essential to nutrition, digestion, etc., because they are the principal emulsifying agent that is used to break up these fat droplets, these big fat droplets. So cholesterol goes through a multi-step synthesis pathway, which is what all those little arrows are meant to represent. It's like many steps, and you can make, there's a variety of different bile salts. The two biggest ones are the glycocholic acid, which is the one in the top box up there, and the one on the bottom, which is the taurocinodeoxycholic acid. Say that quickly 10 times, I dare you, even 3 times. Say it once. Taurocinodeoxycholic acid. Just whip that one out at Thanksgiving dinner. All right. So the process of making a biosalt is essentially the process of putting polar groups on it. So you can see on this thing that is in Wes's hand here, a number of these hydrogen groups, these little white balls that are sticking off of it, get replaced by more of these red and white hydroxyl groups. All right. In fact, we add 2 of them. This one here and this one here. And you'll also notice, if you were looking closely, this one doesn't have the stereochemistry here. But if it did have the stereochemistry, you would notice that those are pointing upwards. They're both on the same side of the molecule. As is this tail here. The isoprenoid tail has been replaced. The isoprenoid tail has been replaced with glycine. And glycine is the simplest amino acid, if you will remember that. So that's also a polar molecule. So we now have this really broadly amphipathic molecule. Not amphipathic longitudinally, but laterally. One side of the molecule, one side of this long, flat molecule, this rigid structure molecule, one side of it is going to be polar, and one side of it is going to be nonpolar, okay? And so it's kind of like an amphipathic patch that you can stick down on a lipid surface and allow it to go into solution. And that's what this cartoon on the bottom is meant to represent. You have this big yellow blob, which represents the bulk of this greasy body of the cholesterol backbone. And then you have these hydrophilic polar groups that have been added that make that side of the biosalt much more polar. We call it a salt because the glycine, the carboxylic acid on the end of the tail, has been ionized, and the counter anion to the carboxylate group there is sodium. All right. So what does this stuff do? It takes a big fat globule that you see on the right-hand side, and it is going to break it up by adding these bile salts onto it, onto the surface of it, and break it up into smaller, nonpolar emulsion droplets. Okay? You guys have any questions? I'm going to move forward to another aspect of this. So I'm going to have to introduce glycerophospholipids. I know this may feel like we're getting in deep here, but don't fear. I'll hold your hand the whole way. A glycerophospholipid is just a triglyceride. It's just a triglyceride where one of the fatty acids has been cut off and it's been replaced with another chunk that is a little bit more polar. Okay? So again, we're just increasing the amphipathic nature of the lipid here. And that thing that's replacing it is going to be choline, and that's the molecule that you see there. Choline is going to, and there's going to be a linker molecule, which is phosphate. Phosphate is a nice bridge between the glycerol backbone and the choline on the end. It doesn't always have to be choline as the polar molecule sticking off the end, but it is choline when we consider lecithin. So lecithin is a molecule, a glycerophospholipid that's found all over the place, and it's typically commercially available through soybean. They extract it out of soybean. But in general, the general structure of a glycerophospholipid is two fatty acids sticking off the bottom of a glycerol, just like any normal triglyceride, except one of the fatty acids is now linked to another polar molecule via this phosphate group. Okay, all that does is increase the amphipathic nature. There's still this greasy tail, but now the head group is much more polar than it would be in a normal triglyceride. You guys following this? Okay, why is this important? Well, it turns out that to make these emulsion droplets out of these big blobs of fat, to make the emulsion droplet, the lipid emulsion, you can't just use bile salts. You also use some triglycerides, and you also use these glycerophospholipids. All right, and we can see here in the cartoon that is on the top right-hand corner, you have the fat, a little bit of a fat droplet, and it's been emulsified because there are these molecules of bile salts that have been stuck on the surface. But in between them, filling in the gaps, we have the glycerophospholipids and also some triglycerides. So they're called glycerophospholipid because they use glycerol as the backbone. They use phosphate as the linker, and they have lipid chains, fatty acids, sticking off the back end. And that phosphate linker links to some other kind of polar molecule. So bile salts and glycerophospholipids. All right. So we just covered the two foundational concepts of emulsification. That was the glycerophospholipids and the bile salts. And I said there was one other thing that we needed, and that was enzymatic digestion. The protein that does that is lipase. This is it. So just like previously when I talked about saponification and making soap, these triglycerides can be converted into a monoglyceride and two free fatty acids by the action of pancreatic lipase. All right, pancreatic lipase is closely associated with the emulsion droplets. In fact, there's another protein that binds to lipase that embeds itself in the emulsion droplet and acts as an anchor, so that lipase is right there at the emulsion droplet, chucking off free fatty acids left and right. Okay, this is pancreatic lipase. It's going to produce monoglycerides, meaning a single fatty acid attached to a glycerol backbone, and two free fatty acids. You guys following this? Is anybody's eyes going pinwheel yet? It's a lot. I know. Covered some ground. Good stuff, though. You guys are learning here. This is, more than 98% of the people out there eating what they're eating. Okay. So here's a bit of an overview again. But a little bit more detailed. I'm filling in some of the details because you have more of the information to understand it now. Step one is emulsification. And that is the process of taking a big fat globule that's from your diet, something you've eaten, and your body has lecithin, has bile salts, bile acids that are going to break up that big fat globule, associate with the fat, and make these smaller emulsification droplets. I should add into that diagram a glycerophospholipid, shouldn't I? I don't have one there, but it should be in that picture. The next step is fat hydrolysis. So there's going to be this pancreatic lipase. So that's just a lipase that your pancreas has made. Okay? Your pancreas makes this stuff called pancreatic juice. The pancreas itself looks like a little gun or something that is being cradled by your stomach, and it releases this pancreatic juice. We're going to see all that. I'm going to go through the GI tract right before. It's pretty much the next thing we're going to do after lipids here. We'll go through the GI tract. But the pancreas releases pancreatic juice into your intestines, and in it is this pancreatic lipase. It's one of the things that's in the pancreatic juice. Pancreatic lipase, lecithin, bile acids, etc., are in these emulsification droplets. The lipase gets to work, and it cleaves the ester bonds for two of the fatty acids on the triglyceride, leaving us the free fatty acids and the monoglycerides. Now, these tiny little free monoglycerides and fatty acids are going to get together with some cholesterol and various fat-soluble vitamins like vitamin E, maybe some vitamin D, maybe some vitamin A that are in your diet, probably mostly vitamin E, and they're going to form these micelles, even smaller. So there's three orders, there's three layers of size scale here. There are these big, totally macroscopic fat globules. There are these emulsification droplets that the lipase works on, releasing free fatty acids, and then those form tiny little micelles, okay? It's these micelles that are going to be the source of the lipids that our gut is going to absorb, all right? All right. So TAG is just the short name for triglycerides. So triglycerides are also called triacylglycerides. Or TAGs is another name for triglyceride. So TAGs and fatty acids cannot directly pass into from the gut into the bloodstream. You can't go straight into the blood from the gut as a triglyceride. Why is that? Well, they're amphipathic. They're kind of hydrophobic. They don't want to be in water, and your blood is actually mostly water. The glucose is happy as a clam, right? You remember those glucose molecules. When I had a glucose molecule over here, there was a lot more red sticking off of it, wasn't there? A lot more of these polar hydroxyl groups sticking off them. But lipids have a lot of hydrophobic regions, so they won't go directly into the blood. We're going to have to go on the odyssey that will take these triglycerides to our cells where they're needed. All right, that's the job of these lipoproteins. This is the last slide I'll go through. I'm not going to even finish it entirely today. I'll just introduce the concept, and then we'll pick up on it and finish off this lecture on Monday. So a lipoprotein is essentially the last category of lipid that I'm going to talk about, although there are others that we could go into. A lipoprotein is simply another ball-like structure, like the fat droplets, the emulsification droplets, the micelles. It's another one of them that is designed specifically to live in the blood. All right, and they are clusters of cholesterol, triglycerides, fatty acids, other kind of lipids, any kind of greasy molecule that needs to hitch a ride in the blood. And it's going to be surrounded by hydrophilic coating. And the constituents of that hydrophilic coating can be quite complex. We are not going to go through all of the components in the surface of a lipoprotein, although it's extremely important to understand those, but it's way beyond the scope of this class. Just take it for granted that there are all these various proteins which as a category are called apolipoproteins. You see the name there on the little white diagram, embedded apolipoproteins. There are not enough hours in the day for me to cover everything I want to cover. So we'll pick up with that. Good work, guys. That was a serious lecture. Yeah, that was worthy of science students there. You guys did it. Hit you with some real knowledge. Go out into the world and spread it.